So if red color is there, it means that they have a particular disorder or a disease. Genetic disorders means any disorder that occurs because of the genes, right? Alteration in the genes, mutation in the gene. This is a sex-linked recessive disease wherein one of the genes which is responsible for causing this particular disease is inherited from the parent. So the representation of chart showing family history is called as family tree, and that is nothing but called as pedigree. Hello everyone, a warm welcome to this session for class 12 biology. I am Dr. Divya, biology faculty. So in this session, that is for chapter 4, principles of inheritance and variation, which is the last session of this chapter, we will learn about the Mendelian disorders and also the chromosomal disorders. So the genetic diseases that are there, they are classified as Mendelian disorders and chromosomal disorders, mainly because Mendel took into consideration only one character, or one gene, wherein only a single gene was considered. So any alteration in that particular single gene can lead to a number of diseases. So those diseases were placed or were called as a Mendelian disorders, and those diseases which were result of either the addition of a chromosome or a deletion of the chromosome, all such diseases were placed under the chromosomal disorders. Now, before learning the disorders, we need to know about something called as pedigree analysis. So, pedigree is nothing but our ancestors, right from our ancestors, that is our great-grandfathers, grandfathers, then our father, from the child, and it continues. So how can we inherit a particular disease, or how can we inherit a gene that is responsible for a particular disease in the progenies? We can get it by analyzing the pedigree chart. So we need to know some of the symbols that they use in the pedigree chart. So we'll talk about pedigree analysis. So the practice of analyzing inheritance pattern of traits in several of generations of a family. That is, say for example, the grandfather, that is the great-grandfather and the great-grandmother, they have married, then they'll have the children. From the children, again, those children will marry. From that, they'll have the next progeny. It continues. So we can find out like where exactly some gene went wrong. Did the gene was wrong in the grandparents itself, or was it wrong from the parents itself? So all that we will get to know. So we can get to know that from where we inherit a particular trait or a character according to the pedigree analysis that we do. So the representation of chart showing family history is called as family tree, and that is nothing but called as pedigree. So in human genetics, pedigree study is very, very important because it is one of the strong tools which is used to trace the inheritance of a specific trait, abnormality, or a disease. Or say for example, before a person marries also, we can get to know whether that family carries a particular disease or not because of this pedigree analysis and all that. Or else what will happen, the child may also will have to suffer later on, right? So we'll get to know where exactly was it wrong and from whom did we inherit that particular trait, that character, or a particular gene that is responsible for the disease. All that we will get to know under pedigree analysis. So these are the symbols that are used in the pedigree chart. So first one is male. For male, if you can see, they have put a box and it is shaded blue in color. For female, they have put a round circle. It is shaded blue in color. And if the sex is unspecified, that is, we don't know whether it is a female or a male, then they will just put it as a diamond shape. Now, affected individuals. Affected individuals means they are shaded in red. So for males, it is again a square with red filled in. For female, again it is a round with red filled in, and unspecified sex, it is again a diamond with red filled in. So if red color is there, it means that they have a particular disorder or a disease. So the affected individuals are marked like that. Next is mating. Mating between the male and the female. So this is the male and the female. So mating is shown by drawing a line at the center which forms the connectivity between the male and the female. Next is mating between relatives. So sometimes what happens, especially in the Indian tradition, it is a habit wherein the marriage takes place within the relatives itself. So either the groom or the bride will be from a relative only. So if mating occurs between a relative, then It is marked like this. Why? Because they can have the traits, that is, the traits will be there within the family itself. No traits from outside can come. So it is marked like, that is male and female. So mating between relatives. Two lines which connect the male and the female. Next is parents above and children below in order of birth, left to right. So here parents, that is male parent, father, female parent, the mother. Mating takes place between the two parents. Then the progenies will develop, wherein female child will be there, male child will be there. So this symbol, if it is there like this, then it is called as, that is, the progenies are also shown there. Next, parents with male child affected with the disease. Now, parents with male child affected with the disease means both the parents are shown here, they are normal. But what happens in this particular case is, either the father or the mother would have that gene which is responsible for the disease, but the gene would be recessive or suppressed in them. Only when they have a child, no, if it is a male child, that male child can receive the trait and the gene can become dominant in that particular male child and the disease can be expressed. So that is what is shown here. Next is 5 unaffected offsprings is shown like this. That is, all the offsprings, if they are unaffected, it is shown like this. So these are the pedigree symbols that are used. So you will get to know how the symbols will, what it will teach you and all that in the next slides, wherein we'll study about Mendelian disorders. Therein for 2 disorders we will get to know about it. Next, talking about the genetic disorders. So genetic disorders means any disorder that occurs because of the genes, right? Alteration in the genes, mutation in the gene, all that we call it as genetic disorder. So it is classified into 2 types. One is Mendelian disorder and the other one is chromosomal disorder. So in Mendelian disorder and chromosomal disorder, first we'll concentrate on the Mendelian disorder. So here I have put 2 pedigree charts. So there are various Mendelian disorders that are there: phenylketonuria, then hemophilia, color blindness, cystic fibrosis. All these are the different Mendelian disorders which we'll study each of them one by one later on. So first, to understand this pedigree chart. So 2 examples that is taken in the NCERT textbook. One represents autosomal dominant trait and the other represents autosomal recessive trait. So one of the best disease that we can take for autosomal dominant trait is myotonic dystrophy. Myotonic dystrophy. And for autosomal recessive trait, we'll take sickle cell anemia. Sickle cell anemia. So these 2 we will take into consideration. So autosomal dominant trait. So which is carrying the autosomal dominant trait? It is called autosomal. So autosomal means what it means? It means the female will carry. That is why it is called as autosomal, because females will have XX chromosome. Males are allosomal because they'll have XY chromosome, right? So therefore, who is carrying the disease there? It is in the female. So female is this. The female is affected. So I hope you understood. There are 2 words, autosome and allosome. You have studied in the previous session also. Autosome means the female, we have totally 23 pairs of chromosomes, that is 46 chromosomes, right? In that, all the 22 pairs are autosomal, but in the male, they have 1 allosome, right? So in female, XX chromosome will be there. In male, 1 XY chromosome will be there. So here it is understood, this is a autosomal dominant trait. So who will get the disease here? It is the female, right? So the female is having which disease? She is having myotonic dystrophy. And according to the symbols that I showed you, if it is circular, it means it is a female. If it is a square, it means it is a male. Now, if it is a square and if it is blue in color, it means the male doesn't have a disease. If it is round and it is red in color, meaning it is a female, she is having a disease. What is the disease? She is having myotonic dystrophy as an example here. Now, what has happened? We have drawn a line in the center between these two. It means mating has occurred between the two. So when mating has occurred, what will happen? They'll produce progenies. Now, what are the chances of them producing progeny? They can produce a girl child who is normal without any disease. They can produce a male child which has acquired the disease. They can again produce a male child which is not having any disease, and they can produce a female child which is having a disease. So these are the possibility of the F1 generation which they produce, or the offspring which they produce, which may carry the disease or which may not carry the disease. Now what we'll do is, what they have done here is, say for example, the next generation, that is the F1 generation is there. If the male child who is not having any disease marries a female who is not having any disease, then the resulting offsprings, none of it, that is, be it the male or the female, they will not have any disease. So that is what it means. Now, in another possibility, they also have, if the female child who is having the disease marries a male child who is not having the disease, now it is a autosomal disorder, right? So therefore the dominant trait will be in who? It will be in the female. So therefore again the female only will receive the disease, not the male. Why not the male? Because the male will have X and Y chromosome. They will not have XX chromosome. So that is why. So like this is how pedigree analysis is done. Now what did we get to know? We got to know that from the affected female, when the affected female and the normal male, healthy male married, the disease passed on to the female child. So that diseased female child, even though she marries a healthy male, again, and if the children that are produced are males, then the male will not be affected by the disease. Only the female child will be affected by the disease. So therefore, this child, from where did she get the disease from? She got the disease directly from her grandmother. That is, this is the child now, this is the mother. Mother got it from the mother, and again this child got it from her mother, right? So that is how we do the pedigree analysis. Next, talking about autosomal recessive trait. Now, if it is recessive, who will get the disease? We will look into. Now, there is the male and the female. Now this is again autosomal, okay? It is autosomal. This person carries the gene which causes sickle cell anemia. The mother that is there, she will carry the gene which causes sickle cell anemia. But the disease, the gene will be there in her body, but she is not diseased. Why? Because the gene is recessive. It is staying calm, not expressing any disease. So therefore the mother is healthy, but still the gene will be there in her body. Now, the mother who is having the gene that is responsible for producing the sickle cell anemia, but she's only having the gene, but the disease is not there. She marries with the normal healthy male, then the resulting progenies can be different, like this. Say for example, they can produce a healthy male child. They can produce a male child who is affected by sickle cell anemia, and they can produce again a female child, healthy female child. They can produce a female child who is affected by sickle cell anemia. Now here again they can also produce a child, female child, who is having the gene. So I'll write it G. She is also having the gene. This one D, meaning diseased, okay? This one also D, meaning diseased. To make you understand, I'm just writing this. So this means, when I write geno, it means this is a female who is having the gene but not having the disease. Because the gene is being suppressed there, it is recessive. Now, possibility is that the male child will get the disease, meaning the gene is also there, the disease is also there. And one of the girl child will also get the disease, meaning the disease is there, gene is also there. Here, this one of the girl child, she got the disease, she got the gene, but the disease is not being showing up because she got the recessive gene here. So that is why it's not getting expressed. Now, when she marries a normal healthy individual, what might happen is, when they have a male child, what happens? The male child will become affected. So therefore, who is responsible? Here, not the diseased person, but the one who is carrying the gene of sickle cell anemia is responsible for the disease in this particular male child. So these are two examples to show you how the pedigree analysis chart, how it comes to use, so that we can find our ancestors from where exactly the disease came from or disease began from. So this is about 2 examples of Mendelian disorders. Next, we'll talk about the remaining Mendelian disorders that are there. First one is hemophilia. So the word hemo means something related to hemoglobin, right? It has something to do with the hemoglobin. So this is a sex-linked recessive disease wherein one of the genes which is responsible for causing this particular disease is inherited from the parent. Now in this, a protein involved in the blood clotting is affected. So which is that protein? It is the heme protein. So we all know that in our blood we have a protein called as heme, and that not just gives color to the blood but also it is responsible for clotting of the blood. Now that protein that is there, it will be absent, it will get affected. Why? Because the gene that is responsible for producing that protein which is responsible for clotting of the blood is no longer functioning properly. And that gene, from where did they receive? They received it from the parent. Now a simple cut. So what happens during hemophilia? One small cut, a paper cut also if you have, and bleeding starts, the bleeding will not stop very fast like other people. So that is what happens here. That is non-stop bleeding occurs with the person having this particular disease. Here, who is the carrier? The carrier is a heterozygous female for hemophilia, may transmit the disease to the son. So from where? From the female. So this is the diseased female. If she marries a normal male, then, so I'll just put this line. So this is a diseased female. Next, the progenies, if they have, say for example, they'll have 2 sons and 1 girl. If they have, then these sons that are there, they will get affected. And so always the carrier is this female. So females transmit the disease to their son. So any female having this particular disorder, it is better she go in for some assisted reproductive technologies wherein the ova can be taken from some other person and she can go in for surrogacy also. So all that options can be adopted instead of having the child through natural birth. So likewise, this pedigree charts and all this help us to understand or helps us to act for what the future holds us. So that is what it will help us to. So here, normal blood vessel, hemophilia blood vessel, it is ruptured. It is a ruptured blood vessel wherein that is during hemorrhage and all that clotting will occur. So that is in a normal person. So when the blood vessel ruptures during hemorrhage at that time, or when we have a wound or something, clotting will occur. But in a normal blood vessel, that is when hemorrhage occurs, if there is a particular gene that is not capable of producing the proteins which is responsible for clotting of blood, then the clotting of the blood will not happen and continuous bleeding can occur. So this is about hemophilia. Next, talking about color blindness. Color blindness is the inability to distinguish between the colors or see the colors. So it is also a sex-linked recessive disorder because it is got from the parent, and it is caused due to defect in either red or green cone of the eye. So the cones in the eye are responsible for perceiving the color. So anything wrong with that cone of the eye, that is the red or green cone of the eye, it will result in the failure of discriminating between the red and the green color. So this defect is usually caused due to mutation in certain genes present in the X chromosome. So in the X chromosome, if there is any defect in the gene that is responsible for maintaining the cone of the eye which is responsible for detecting red and eye color, then the person will become color blind. So here also one gene is responsible. There also one gene was responsible for the disease in hemophilia. That is gene responsible for production of the protein. So that is why it is called as Mendelian disorders. One gene is responsible for the disease here. So that is why. Next is thalassemia. So thalassemia, it occurs either because of mutation or deletion of that particular gene, which actually results in the reduction of synthesis of globin chains that make up the hemoglobin. So hemoglobin is wherein we have two components in the hemoglobin, right? That is heme component and globin component. Now, if there's something wrong with the globin component or the globin chain, then this particular thalassemia disease will occur. So, thus an abnormal hemoglobin molecule results in anemia, and based on that we have alpha thalassemia, beta thalassemia and delta thalassemia. So, these are the different types of thalassemia that is there. So, what happens here? As you can see, normal blood and you can see thalassemia blood. Normal blood, they'll have the red blood cells, the white blood cells, platelet, all that is there. In thalassemia, you can see the malformed red blood cell because that globin chain that is there, that gets affected. So, therefore, the shape of the heme protein will change. So, therefore, the shape of the red blood cell will also tend to change here. So, malformed red blood cell, all that will happen. You can see here normal blood, they'll have somewhat concave shape of RBCs, but in this case you can see oval. So, all these are oval shapes or different shapes of RBCs. If you can see here, it is because of the problem in the globin chain of hemoglobin. Therefore, the hemoglobin content only will become a problem, causing disfigurement of the red blood cells or malfunctioning of the red blood cells. So, this is about thalassemia. Next, sickle cell anemia. The name itself says sickle cell. Here, the red blood cells, which are supposed to be oval or concave in shape, they become like a sickle. So, sickle is almost like this. They become like a sickle. So, usually red blood cells, the shape is like this. That is normal RBC. This is sickle cell RBC. So, this is how it looks. So, this is an autosome linked recessive trait. It can be transmitted from parents to the offspring when both the parents are carrier of the gene for heterozygous. So, here either the father or the mother, anyone, if they have this particular gene which is responsible for changing the shape of the RBC or making the RBC sickle shape, they can transfer that particular disease to the offspring. So, the disease is controlled by a pair of allele, that is HB and HD. So, alleles are nothing but they are present on the gene itself. So, they are HB and HD. So, here what happens we will look into. So, they have given here normal hemoglobin gene and sickle cell hemoglobin gene. So, normal hemoglobin gene, say for example, we have GAG and GCT. So, it's just like that. This is the DNA strand. So, DNA, they are double stranded. So, two strands are there wherein lot of genes will be present. In these number of genes, what are our concern? We are concerned about only GAG gene and CTC. That is complementary because G complements C, A complements T and G complements C. All this you have studied in your previous classes. Now here, what happens here is next. So this, now an mRNA strand will be produced for this. So this is the mRNA strand that gets produced for this. Now in the mRNA strand, what should they have? They should have again complementary, that is G. So for this mRNA strand is being produced, right? So always remember in DNA, adenine base pairs with thymine and guanine base pairs with cytosine. In RNA, adenine base pairs with uracil, guanine base pairs with cytosine. They don't have a thymine. So now here I am drawing an mRNA strand. So for T, can I write a here? No, because they don't have. I can write a because they have adenine. But if it was A here, I should have written U, right? So here GCG or GAG. Now this GAG that is there, it is responsible for producing all of these that are there. Valine, histine, leucine, all these which are there, they are responsible for production of various amino acids. So here what happens here is this GAG that is there on the mRNA strand is responsible for production of the amino acid glutamine or glutamic acid. So what does it produce? It produces glutamic acid. Now this glutamic acid that is there, it should produce only glutamic acid. That is what happens in the case of a normal hemoglobin gene. But in a sickle cell hemoglobin gene, what happens here is, in the DNA, one of the DNA strand, they have GTG, and the opposite DNA strand, it should be CAC. It will have CAC. Now, actually, if it was a normal hemoglobin gene, the gene should have been GAG, right? But here there is a mutation. What happened? Instead of A, T came in that place, GTG. Now, if GTG happens, what happens in the complementary DNA strand? It will be guanine for cytosine, thymine for adenine, guanine for cytosine. Now, when an mRNA strand gets produced for this particular strand of DNA, what happens? Cytosine for guanine, for adenine it is uracil, for cytosine it is guanine. It became what? GUG. Now, GUG will not produce glutamic acid. Glutamic acid is produced by which gene? GAG. So here GUG, what will get produced? Valine will get produced. Now, when this valine gets produced, what happens? It will alter the production of the protein. Therefore, what will happen? The RBC cells that are there, they will start becoming sickle cell instead of being oval or concave shape. So, see how one small gene that is there, it is very important, or one small allele that is there, how it is very, very important, right? So, this is about the sickle cell anemia. Moving further, we'll talk about phenylketonuria. So, phenylketonuria, if you remember, this was an example for pleiotropy, right? We had studied in the previous session. Pleiotropy, it is an example. Pleiotropy means one gene controls many characters. So, the same thing we'll study about this here. It's also a Mendelian disorder because one gene is responsible for, the alteration in one gene is responsible for phenylketonuria. So, phenylketonuria, it is an inborn error, that is, in the fetus itself, this error will develop, and it is an inborn error of, or it is a birth defect. It is an inborn error of metabolism, and it is an autosomal recessive trait. It is autosomal recessive trait means it is got from the mother. It is got from the mother. The affected individual will lack an enzyme. That enzyme's name is phenylalanine hydroxylase enzyme. And this phenylalanine hydroxylase enzyme is required to convert amino acid phenylalanine into tyrosine. So, in our body, whatever amino acid, that is phenylalanine is an amino acid. It should get converted into what? It should get converted into tyrosine. For the conversion of this phenylalanine amino acid into tyrosine, there is one enzyme that is required, that is called as phenylalanine hydroxylase. This enzyme is required. Now here what happens, and this is produced, and this phenylalanine hydroxylase enzyme is produced by one of the genes that is present in a DNA or present in the chromosome. Now this gene, if it gets altered or if it gets mutated, enough amount of phenylalanine or phenylalanine hydroxylase enzyme will not get produced in the body. And if this particular phenylalanine hydroxylase enzyme is not produced in the body, phenylalanine amino acid will not get converted into tyrosine. Therefore, what will happen? Phenylalanine will get accumulated in the body, and that phenylalanine will get converted into phenylpyruvic acid. Now, if phenylpyruvic acid gets accumulated in the brain, it will result in mental retardation. Therefore, it is a disorder related to the brain. Which gene controls it? Only one gene that produces the phenylalanine hydroxylase controls the phenylketonuria. So, this is about phenylketonuria. Moving further, we'll talk about the chromosomal disorder. So, chromosomal disorder can happen because of an addition of a chromosome or a deletion of the chromosome in the 23 pairs of chromosomes that we have. So, they are caused due to absence or excess or abnormal arrangement of one or more chromosomes. They are of two types, aneuploidy and polyploidy. Aneuploidy is nothing but loss or gain of chromosomes. It can be either loss of one chromosome or gain of one chromosome. It is called aneuploidy. Polyploidy means many. So, here a set of chromosome increases in number. That is called as polyploidy. So, we'll see about this. Aneuploidy. Aneuploidy, the gain or loss of chromosome due to failure or segregation of chromatids during cell division is known as aneuploidy. So, during cell division what happens? The chromosome segregates and they move into the daughter cells. Now, during the segregation process, if equal amount of chromosomes doesn't get distributed properly, then what will happen? An extra chromosome will get added up there. So, therefore, it can be lost or it can be gained. So, that is called as aneuploidy. So, some of the examples of aneuploidy are Down syndrome, Klinefelter syndrome and Turner's syndrome. So, let's study about each of these one by one. So, talking about Down syndrome. So, Down syndrome is a genetic disorder. It is caused due to the presence of an additional copy of chromosome number 21. So, we have many number of chromosomes, right? We'll start counting. How many pairs do we have? 23. So, we'll start counting. This is the first pair, second, third, then fourth, 5, 6, 7, 8, 9, 10, 12th one, 13th, 14th, 15th, 16th, 17th, 18th, 19th, 20, 21, 22, 23 pairs. 23rd pair is X and Y. They have shown here. So, 23 pairs of chromosome is there. So, here what happens? In the 21st chromosome, if you can see here, everywhere they are in pair, right? But here how many is there? 1, 2, 3. Therefore, trisomy condition, that is, there is an addition of the extra chromosome in the 21st pair of chromosome. So, that is called as trisomy or number 21 trisomy results in Down syndrome. And this disorder was first described by Langdon Down. So, that is why it is called as Down syndrome. So, here it is represented like this. So, we'll take this into consideration. So, wherein 45A plus XX. So, why it is written like this means 45 are autosomes there, right? All the 45, that is up till here. 1, 2, 3, 4, 5 till here. That is till all these, if you can see here 2, 4, 6, 8, 10, 12. So, if you count all the chromosomes, there are 45 totally and all are autosomes wherein one allosome is there. So, this one, the 23rd one is allosome. So, therefore, plus 1 allosome which they have represented as X and one more chromosome which got, that is 21st. In the 21st, they got one more extra chromosome, no? So, that plus 45, 46, 47. So, a Down syndrome patient or person will have 47 chromosomes totally instead of 46. So, next, Klinefelter's syndrome. So, it is a genetic disorder caused due to the presence of an additional copy of X chromosome. So, they have an additional copy of X chromosome. So, additional copy of X chromosome means they will be in the allosome. It occurs in the allosome. So, this one, if you can see here. Here, one extra. It was supposed to be X and Y chromosome, but instead, one more X will get added up here. So, that is called as Klinefelter's syndrome. And the genetic constitution will be 44A plus XXY. So, why? 44 autosomes are there. 44 autosomes are there. And here they have 2 X chromosomes. How many? 1 plus 1. 2 X chromosome and 1 Y. So, 44, 45, 46, 47. Totally 47 chromosomes they will also have. That is here. So, 44 up till here. Okay, here 44 if you can count. 22 pairs means single 44 chromosomes. Here 45, 46, 47. So, totally 47 chromosomes they will have. So, that is Klinefelter's syndrome. Next is Turner syndrome. So, both Down syndrome and Klinefelter syndrome, both there was addition of chromosome. In Turner syndrome, there is absence or deletion of one of the X chromosomes. So, absence or deletion of one of the X chromosome, that is here. Here there are no chromosome, that is absence or deletion of one of the X chromosome. So therefore genetic constitution is 44A plus XO. So here you can see they will not have one X chromosome, or here also there is no X chromosome at all. So there is absence or deletion of one X chromosome. So that is why it is represented as 44 plus 1. So this till here it is 44 and they'll have one more here. So 44 plus 1. 45. 46th one, it is deleted. 46th one got deleted. So therefore, it is 44A plus X0, that is 44 autosomes plus 1 X chromosome. 45 chromosomes will be there. And the females are sterile as ovaries are rudimentary and they lack secondary sexual characters. So the females who are having this Turner syndrome, they will usually be sterile, because they will not have one X chromosome in them, which is responsible for the production of the ovaries as well. So next, moving to polyploidy. So in polyploidy, it is an increase in a whole set of chromosome due to failure of cytokinesis after telophase. So after telophase, what happens? The cytokinesis, or during cytokinesis, what happens? Either the division of the cells, wherein the cell separates to form two daughter cells, that will not happen. So when that doesn't happen, all the chromosomes that is there, 46 plus 46 for example, everything will get constrained in one cell itself. So that is called as polyploidy. Multiple number of chromosomes will get accumulated in one cell, because the cytokinesis, or the actual stage, that is last stage of cell division, wherein the two daughter cells are supposed to form, that will not get formed. And this condition is often seen in plants. So that is why strawberries and all that, they will have multiple seeds in them, if you can see. So strawberries are one of the examples. So they have haploid, then diploid chromosomes. Some of the cases there are triploid chromosomes. They have tetraploid chromosomes. All these, each two or the chromosome should have got equally separated to the daughter cells. But because cytokinesis itself does not occur, all the chromosomes will get accumulated in one single cell only, which is nothing but called as polyploidy. So this is about the different chromosomal disorders, as well as we studied Mendelian disorders also. So I hope you found this session useful. So this was the last session of this topic, that is of this chapter. So we shall meet again in the coming session, wherein we'll discuss the topics under the new chapter. So see you in the next session. Thank you.